Okay, so today we will talk about mainly the muscle contraction, the mechanism of muscle contraction. But we'll also talk about a few other things. First, we'll see three different types of muscle. You already know it. Then we'll talk about the functions of the muscles. Then we'll talk about the structure of the skeletal muscle. Then we'll talk about a very important structure which is like a filament, thin rod-like structure that is called myofibril. Inside the skeletal muscle you have myofibrils, those thin filament or rod-like structures. Then we'll talk about the contractile units. Contractile units include the contractile proteins and thick and thin filaments. So we'll see who are the contractile proteins and thick and thin filaments. Then we'll talk about a couple other things: cross bridging, neuromuscular junction, and clinical conditions. So basically, we will talk about the skeletal muscle. Now, three types of muscle tissue are present in the body or found in the body. You already know: skeletal muscle tissue, cardiac muscle tissue, smooth muscle tissue. Cardiac and smooth are involuntary. That means we cannot control them, right? You cannot stop your heart or control your heart. You cannot control your smooth muscle, which forms the tube-like structures, right? Blood vessels or esophagus or trachea. You cannot control them. So, involuntary. But skeletal muscle, you can control. Skeletal muscles are attached to the bones, right? And you can move the bones as you wish. So, only skeletal is voluntary. Now, how they look like under the microscope, you have already seen these different types of muscle tissue under the microscope. Just quick review. This is a skeletal muscle tissue. The fibers are cylindrical. Striations are present, right? And multiple nuclei. In each fiber, you have multiple nuclei, and nuclei are not located in the center but attached to the membrane. Peripherally located multiple nuclei. Okay? So that is the skeletal muscle tissue under the microscope. If you see the cardiac muscle tissue, fibers are cylindrical too, striations are present, but in case of cardiac, the striations are not that prominent like skeletal. In skeletal, more clear. Here, less clear, but striations are present. Another important thing here, which is not present in skeletal, intercalated discs. These thick lines you see, bands, intercalated discs, only present in cardiac. And another thing you have in cardiac, which is not present in skeletal, is branching. Okay, so that's the cardiac muscle tissue under the microscope. If you see smooth muscle tissue, smooth muscle cells are spindle shaped. Both ends are pointed. Single nucleus located centrally. No striation, no intercalated disc, no branching. So very simple. Spindle shaped fiber, centrally located single nucleus. Okay. So many smooth muscle fibers or cells join together to form the tissue, and it looks like this. Okay. Sometimes it is difficult to see the membrane, and that makes it difficult to identify, but this is actually how the cells are together. Okay. So this is smooth muscle. So those are three types of muscle tissues. Now let's see the properties of muscle tissue. How the muscle tissue is different than other tissues of your body, right? Muscle tissue has some properties. What are those special properties or characteristics? Excitability, the ability to respond to a stimulus. If I poke your muscle, right, it will respond. So, if you stimulate, the muscle will respond to the stimulus. Make sense? That ability is called excitability, the ability to respond to a stimulus. Now, contractility. How the muscle responds? By contraction, right? By showing contraction, shortening. Is it clear? Shortening is contraction. Extensibility. If I hold the muscle tissue like this and pull from both ends like this, it will expand, stretch, right? And that ability to elongate is called what? Extensibility, right? Now you look at me. After I expand the muscle by pressure, if I release that muscle, what will happen? It will go back to its actual shape, right? So that is called elasticity, recoiling. You know, elastic thing you use in your clothes, right? You can expand, that's the expansibility. Now if you remove the pressure, it will go back to its actual shape, right? Shortening. So that's the recoil, elasticity. So those are some special properties of your muscle tissue. Functions. Movement. Skeletal muscles are attached to the bones, and by the contraction and relaxation of the skeletal muscles, we move our body parts or bones. Circulation of body fluid. You know, your heart pumps the blood, right? Heart is cardiac muscle, and by the pumping action of your heart, blood circulates throughout the body, right? 24 hours your heart is pumping the blood, and that is causing the circulation or flow of blood. Maintaining posture and body position. By using our muscles, we maintain the body position. Stabilizing joints. In last class, I mentioned that muscles cross the joints, right? From different sides like this, and some muscles prevent, you know, undesirable movement, right? And so stabilize the joints. Unnecessary movement is prevented by the muscles because they cross the joint. Heat production or generation of heat. You all know, we have talked about this before, right? When hypothermia, hypo means what? Low, right? Thermia means temperature. So hypothermia or body temperature lowers, goes down, shivering occurs, right? Your skeletal muscles contract in high frequency. Why is that? We know that contraction of muscle will produce heat, right? Metabolism increases, that produces heat. Is it clear? So by increasing metabolism, your skeletal muscles produce more heat to increase the body temperature in hypothermia. Is it clear? So that's how your muscles regulate the body temperature. So those are the functions. Now let's talk about the structure of the skeletal muscle. First, if you see the whole muscle spindle, for example your deltoid, the whole muscle, okay? So this is the whole muscle spindle, okay? If I cut it, what I'll see inside? I'll see structures inside it, columns. So basically like this. Many columns are together, right? Make sense? So if I cut the muscle, whole muscle, I'll see the columns like this inside. And each column is a fascicle. Okay, so these are fascicles. Okay, these are fascicles inside the whole muscle. Now if I see inside the fascicle, what I'll see again columnar structures. Okay, and these are called muscle fibers. Muscle fibers. Okay. So inside the whole muscle you have fascicles. Inside the fascicles you have muscle fibers. Is it clear? Okay. So now inside the muscle fiber, again, if I see inside a muscle fiber, I'll see many filaments, thin rod-like structures. Okay. Thin rod-like structures. Those are called myofibrils. Okay. So myofibrils. So you see, this is the structure of the skeletal muscle. In one sentence, I can say what? Columns within columns, right? So within big columns, you have smaller columns. Make sense? All those are rod-like or columnar structures. So this kind of arrangement gives the muscle flexibility and strength. Make sense? Strength and flexibility. Columns within columns. Some tall buildings are built based on this idea. You know Sears Tower in Chicago. It's a very tall building, right? The structure is columns within columns. Smaller columns within big columns. Okay? So that gives the strength at the same time flexibility. Anyway, so this is whole thing is what? The muscle, right? Muscle spindle. Inside the muscle you have many fascicles, right? Inside the fascicles you have many fibers. Inside the fibers you have many myofibrils. Is it clear? So large to small. And myofibrils are what? I said filaments. Thin rod-like structures, okay? Are myofibrils. Now we'll see the covering. The whole muscle is covered by a connective tissue membrane like this, and that is called epimysium. Epimysium covers the whole muscle. Then each fascicle also has covering. So each fascicle is covered by connective tissue membrane, and that is called perimysium. Okay, perimysium. So this is perimysium. Each muscle fiber is covered by, again, connective tissue membrane, and that covering of muscle fiber is called endomysium. Endo means inside. Endomysium. So those are the coverings. Whole muscle is by epimysium, fascicles by perimysium, muscle fibers by endomysium. Okay? So those are three coverings. That's the structure of the skeletal muscle. Now let's see this picture. This is the whole muscle spindle. Okay, if you cut it, you will see many fascicles inside. This is just one fascicle they have taken out. And you see the whole muscle is covered by epimysium, fascicle is covered by perimysium. So one fascicle is this one. So this one is this whole thing covered by perimysium. And inside the fascicle, you have muscle fibers, right? Now if I just see one muscle fiber, this is the muscle fiber covered by endomysium, okay? Now, this is very important. Inside the muscle fiber, you have myofibrils, right? Thin rod-like filaments. So, you see the muscle fiber, this is the muscle fiber filled with myofibrils. Okay? Many myofibrils or thin filaments. So, now if I see here, again same thing, this is the whole muscle, fascicles, this is one fascicle, and then this is a muscle fiber, and inside the muscle fiber you see rod-like structures, myofibrils. So these are myofibrils. Okay? Now if I take one myofibril out from there, if I take one myofibril out and see under the microscope, what would I see? So I just took out one myofibril. This is a myofibril. Myofibril, okay. Inside the myofibril, you will see segments like this. Same thing is repeated. So several segments. Each segment is called a sarcomere. Sarcomere. So this is one sarcomere, this is another, okay. So inside the myofibril we see several sarcomere. Each sarcomere is the functional unit. Functional unit. That means unit for muscle contraction, right? Function of muscle is what? Contraction, right? So, why sarcomere is called the functional unit? Because muscle contraction starts from the sarcomere. What happens you see here, when muscle contraction occurs, all this sarcomere within myofibril get what? Shorter, like this. Okay? So now think that all sarcomeres get shorter, so whole myofibril gets shorter. Make sense? And inside the muscle fiber, you have many myofibrils, right? Filled with myofibrils. So when all these myofibrils get shorter at the same time, whole muscle fiber gets shorter. Make sense? And when all muscle fibers inside the fascicle get shorter, the fascicles get shorter, right? And when all the fascicles inside the whole muscle get shorter at the same time, that is the muscle contraction. The whole muscle gets shorter. Is it clear? So, where this shortening starts? At the sarcomere. Is it clear? So, sarcomere is the unit of muscle contraction, the smallest unit of muscle contraction. So, but it is important that all should get shorter at the same time, not different times. Okay. Now, if I see a sarcomere under very powerful microscope, like electron microscope, if I see a sarcomere, inside the sarcomere you have filaments. Again, filaments. Two types of filaments are present inside the sarcomere. Thick filaments and thin filaments. Okay. So, these are thick red are thick filaments and the green are thin. Thin filaments. Okay. So two types of filaments inside the sarcomere. Thick filaments and thin filaments. Now, if I see how the thin filaments are attached, you see here, these are called Z discs. Z discs separate the sarcomeres. So the sarcomere is the area between two Z discs. Okay, so these are Z discs. So between two Z discs is the sarcomere. Now you see the thin filaments are attached to the Z disc. So one end of thin filament is directly attached to the Z disc. The other end is free. Okay? So this is the free end. This is the attached end. Now, how about thick filaments? Thick filaments are attached to the Z disc by spring, protein spring, spring-like protein structure, and that is called titin. T-I-T-I-N. So, protein springs attach both or connect both ends of thick filament to the Z disc. Make sense? So, the protein springs are called what? Protein springs are called what? Titin. Okay. So, the thick filament, both ends are attached to the Z disc by titin. But thick filament, one end is directly attached to the Z disc, other end is free. Is it clear? Okay. Now, I'll see more microscopic. I'll see the structure of a thin filament first. If you see a thin filament is formed by double chain like this. It is a double chain structure. So, two chains are formed by these balls. You see round structures. These are called actin. So, actin. Many actin balls get attached to each other to form how many chains? Two. Okay? So, double chain structure is thin filament. So, this is thin. Is it clear? Okay. On the actin balls, you have the binding site. All the actin balls have binding sites. These are called site for myosin. Okay. So on the actin balls, you have site for myosin. Okay. Binding sites for myosin. So that is the thin filament. Now we will see the structure of a thick filament, the red one. The thick filament is formed by a number of golf club-like structures. Have you seen golf club? Okay. So, many golf club-like structures bundled together to form the thick filament. So, this is thick filament. Now, each golf club-like structure or unit of thick filament is called myosin. So this is myosin. Each golf club, okay, is a myosin. So I can say this is what? Actin, single actin, right? Unit. And this is what? Myosin. Is it clear? So many actin balls join together to form thin filament, right? And many myosin bundled together to form what? Thick filament. Is it clear? Okay. Now, myosin has a head part. This is the head. Makes sense, right? Head part. And this is called tail. Okay? This makes sense. So, myosin unit has head and what? Tail. On the head, you have two binding sites. One is a site for actin. Now you see here, on actin you have site for what? Myosin. And on myosin you have site for actin. Make sense? So, site for actin. Is it clear? Okay. There is another site on the myosin head that is called site for ATP. You all know ATP, right? Gives what? Energy. Okay, good. So, now you can think that myosin head has a site for actin, actin has a site for myosin, right? So, they supposed to get attached to each other, right? Supposed to get attached to each other. And during muscle contraction, that happens. That must happen, okay? To contract the muscle. But there is a problem. Problem is the binding sites on actin ball are covered by a rope-like structure. So you see, this rope-like structure is covering the binding sites on actin. Is it clear? And this structure is called tropomyosin. So tropomyosin is a rope-like structure that lies on the actin balls. Is it clear? And covers the binding sites on the actin ball.
Rope-like structure.
Yeah, on the actin balls, right? Now, another thing is on tropomyosin, you have small protein structures like this. Remember, all these are protein structures. And these tiny protein structures that are attached to the tropomyosin or attached on that rope, these are called troponins. Troponins. Okay? So these are troponins. Got it? Okay. So now we see what happens. The myosin head must bind to the actin ball. Okay, must get attached to the actin balls, right? But the problem is what? This, right? It is covering the actin balls, right? So myosin head cannot get attached. Okay, and that is the resting condition, relaxed condition, when your muscle is relaxed. Is it clear? Now when the muscle will contract, the myosin head should get what? Attached to the actin balls, right? Binding should occur. So now you can tell me to free the binding sites on the actin balls. If you want to free them, what you have to do? You have to push the tropomyosin away, right? Move it away. Move the rope away. Is it clear? So how that happens? You see, to do that, you need calcium. Calcium is essential for muscle contraction. Okay? You need what? Calcium. So calcium is released here. And the calcium molecules get attached to the troponins like this. So that is calcium. Calcium ion. Okay? Is it clear? So when the calcium ions get attached to the troponins, what happens? All these troponins move to the side like this. All the troponins move, okay, like this. So now you see the troponins are attached to the tropomyosin, right? Sitting on the tropomyosin. Many troponins, right? So when all of them move like this to the side, it moves the tropomyosin to the side, right? Move the tropomyosin. So that frees the binding sites on actin balls. Is it clear? Okay. So now let me draw here. So this is the myosin head, actin ball. This is the site for myosin head. This is the site for actin. And there is another site, that is the site for what? Site for what?
Complex.
ATP. Right? Okay. And you have tropomyosin here. Now, these are troponins. Calcium gets attached. So these are calcium molecules, and troponins will move to the side. That will move the tropomyosin away. Push the tropomyosin to the side. Now the binding site is free, right? So what will happen? The myosin head will come down and will get attached to the actin. Make sense? Now it is attached because they have sides for each other. Clear? And that is called, that attachment is called cross bridging. Cross bridging. Okay, that attachment. Now, after that, what will happen is, see, this is interesting. You have a binding site for ATP here, right? So, ATP will come and will get attached here, ATP. ATP gives what? Energy, right? So, ATP will come to give energy. Is it clear? So, this is cross bridging attachment, and ATP will come and will give energy to move the myosin head like this. That is the flexion of myosin head. Make sense? So this will happen. Now, after the flexion of myosin head, what will happen next? The head will get detached immediately. Okay? So the head will move this way as well as that way, right? So in both ways you need ATP. Because to move, you need ATP, right? So this is movement, that is movement too, right? So in both flexion and extension, you need ATP. Just remember that, okay? Now, that movement, this movement, flexion of myosin head is called power stroke. Power stroke. Power stroke is the flexion of myosin head. So first cross bridging, then power stroke. Is it clear? This is what? Attachment is what?
Cross bridging.
Yeah, then flexion of the head, power stroke, okay? Now, you see here, actually what happens, once the rope or tropomyosin is pushed away, all myosin heads get attached at the same time like this. Make sense? Million of myosin head. All these are very microscopic, right? So, million of myosin heads get attached to the actin balls, right? Million of actin balls at the same time and do power stroke at the same time. Make sense? So now you can think that it will give a lot of power, right? So that causes the shortening of the sarcomere. Why? Now you see here, these are myosin heads. These are myosin heads. Get attached to the actin balls. Myosin heads. Myosin heads. Same way here. And we'll do what? From this side, these myosin heads will move this way, and from this side, these myosin heads will move that way. Make sense? Like this. So, we'll pull the thin filaments towards each other. That means towards the center from both sides. Make sense? So, since the thin filaments are attached directly to the Z disc, so they will move the Z discs towards each other. Make sense? Like this. So that is the shortening of the sarcomere. Shortening of all sarcomere will occur at the same time that I mentioned, right? So that will will cause the shortening of the myofibril. When all myofibrils will get short, the muscle fiber will get short. When all muscle fibers will get short, fascicles will get short, right? When all fascicles will get short, whole muscle will get short. Make sense? So that is the mechanism of muscle contraction. Okay? So, okay. So, that's the mechanism. Let me quickly go over. Okay? So, this is a myofibril, right? Thin filament. Inside the myofibril, you have the segments. Each segment is what? A sarcomere, right? Separated by what? Z discs, right? So if I ask you, sarcomere is the area between what? Two Z discs, right? And now, if I see inside the sarcomere, how many types of filaments you have? Two, thick and thin, okay? Thin filament, one end is directly attached to the Z disc, right? Other end is free. Thick filaments, both ends are attached to the Z disc by spring-like structure, right? Protein springs called what? Titan. Okay? Now, if you see a thin filament is formed by what? Two chains. And chains are formed by actin balls, right? It's like necklace, beads. And actin ball has a binding site for what? Myosin, right? Myosin. Okay, so that is the thin filament. Now if you see thick filament, it consists of many myosins, right? And each myosin has a head, actually double-headed. So just know each myosin had head and a tail. Is it clear? Okay. And on the head, you have site for what? Actin, as well as site for ATP. Okay. So myosin head should get attached to the actin balls, right? But who is the problem? That rope-like structure called tropomyosin. Make sense? It covers the actin balls, right? Binding sites on actin balls. That is the relaxed condition of the muscle, right? Should stay separate. Myosin head and actin ball should stay what? Separate. Now, on tropomyosin, you have small protein structures. Those are called troponins, right? Now, to move the rope to the side, tropomyosin to the side, you need which chemical? Calcium ion, right? Will get attached to troponins, right? We'll move the troponins to the side, so that will move the tropomyosin to the side, right? So the binding sites on actin balls will get free, open, right? So now the myosin head will get attached, right? So that attachment is called what? Cross bridging, right? And then ATP will come, get attached to the head of myosin, right? And you know that ATP gives energy to do what? Flexion of the head. Make sense? And that is called power stroke. Okay. Have you seen rowing boat? You'll see big long boats. Many people hit the water with paddle at the same time, right? Like in boat race, they will hit at the same time, right? So same thing happens at the same time. Myosin head should get attached to the actin balls, right? Do like this and get released. Otherwise, it will not create power, right? So to produce power should happen at the same time, right? Million of them will do that. Okay. So that's the mechanism. Okay. Okay. See, I'm quickly now going. So this is a Z disc, this is a Z disc. So this part is a sarcomere. Make sense? Between two Z discs. Now you see the blue lines are thin filaments and red lines are what? Thick filaments, right? And you see here more clearly. You see this is the thick filament. These are thin filaments. Okay? And tightening, spring. Okay? Make sense? You see tropomyosin, that rope, and troponins. Okay? This is the myosin, head and tail. Okay? This is the thick filament. This is thick filament, actin ball, binding site. Okay. Now, you remember I said calcium is released, right? And calcium binds with troponins, right? To move the troponins. From where the calcium comes? Calcium is stored around the muscle fiber. So you see these blue structures, tube-like structures, they are connected to each other. That is called a sarcoplasmic reticulum, or SR. Sarcoplasmic reticulum, or SR. How many of you remember endoplasmic reticulum? ER. Remember, right? Inside the cell, remember that? Around the nucleus you have tube-like structures connected to each other, right? Ribosomes are attached. So rough surface here, smooth surface here, right? So similar structure you see, tube-like structures are connected to each other around the muscle fiber, right? And this is called what? Not endoplasmic reticulum, this is called sarcoplasmic reticulum, okay? And these tubes are filled with calcium. Filled with what? Calcium. And you remember that calcium is essential for muscle contraction. So what happens when we decide to contract the muscle, we send signal to the sarcoplasmic reticulum to release the calcium into sarcomere. Calcium should get in, right? So that's the structure that stores calcium. Now, how the calcium will get in from the outside? So this is the, again, sarcomere. You have
What happened to your
Sarcoplasmic Reticulum, the tube-like structures here, SR, this is SR, filled with calcium. So calcium inside. What happens when signal arrives, tells the sarcoplasmic reticulum, now you release calcium because we need muscle contraction. What happens? You have a tube that takes the calcium from SR to inside. That is called T-tubule. T-tubule. That takes the calcium from SR to inside.